this course is laid out, and we'll briefly review the history of DNA and molecular biology. So that's what we will do for this lecture. And the objectives for today's lecture are, number 1, define molecular diagnostics. Number 2, give a brief outline of the molecular diagnostics course. And number 3, for each of the following, briefly summarize his or her contribution to the understanding of.
This is something that all of you know, I'm sure all of you know about literally everyone, Gregor Mendel, Frederick Meister, Frederick Griffith, the transformation one, Oswald Avery, who are showing that, DNA is a genetic material, Chargoff for the Chargoff's rule, right? Rosalind Franklin and Morris Wilkins for deducing the X-ray crystallography of DNA, and James Watson and Francis Crick for elucidating the double helical nature. Right? So, but we will go through this.
Molecular biology. And that will include Gregor Mendel, Frederick Meister, Frederick Griffith, Oswald Avery, Colin MacLeod, and Macklin McCarty, Erwin Chargoff, Rosalind Franklin and Maurice Wilkins, James Watson, and Francis Crick. So you may be asking yourself, what is molecular diagnostics exactly? Well, molecular diagnostics refers to the detection of specific DNA or RNA molecules. It is the study of nucleic acids and specific nucleic acid sequences. And there are lots of different test methodologies that will utilize molecular technology and many different applications for molecular testing. Speaking of applications for molecular testing, let's touch on how it is useful in the clinical lab. Applications in the clinical lab can include detecting infectious diseases or cancer. We can test for genetic diseases. We can do histocompatibility testing, and we can do human identity testing, such as in forensics or paternity testing. So there would be no MORI without molecular diagnostics. So as we go through this course, we will discuss many of these applications and different test methodologies. And on the subject of going through the course, here is a brief course outline. So today we are going to cover a brief history of molecular biology. Then over the coming weeks, we are going to move on to basic concepts of molecular, common techniques in molecular testing, what techniques are used in the clinical lab, and finally, we will discuss quality assurance and quality control in the molecular lab.
So, quality assurance and quality control is something that's very, very important, okay? What it does is that it makes sure the results between two different labs are reproducible, right? We can't be having, one lab showing some result, another lab showing another result. It cannot be, okay? So quality control and quality assurance make sure that the test kits, whatever is on the market, meets certain specifications, right? It needs to make sure that the results that are coming out of this is valid. For example, if you remember the professional ethics course, when we talked about, where we talked about, breach of confidentiality, right? When the employee goes out and tells the tests are not coming out what it's supposed to be, right? That's a major red flag, right? You cannot have going into one lab and says that your sugar level is this, and then you go to another lab, it says, no, your sugar level is normal. That cannot be the case, right? So when they do a sugar test, it doesn't matter which lab that you're testing in. No, it has to come within a certain specified range. So this is what is, taken care by quality assurance and quality control.
I think you will find that the molecular lab is a bit different than maybe the core lab or micro or blood bank. So the layout needs to be very specific and the workflow is very specific, and it's important to maintain that workflow. And also quality control and quality assurance can be a little bit different. So we will talk about all of those. But today we're going to start with history. And I know, I'm sure many of you have learned about the history before, but we're going to talk about it again because I think it's important. And why do I think this is important? Well, I'll tell you. So I know what you guys are thinking. History, really? This is a science class. I'm so over it. I don't want to learn history. But I'll tell you a little bit about myself, and then maybe this will make more sense. So this is me, and this is my giant goober dog named Opal. A long time ago, in a galaxy far, far away, I went to college, and I came out the other side with a history degree. Okay, so this is making sense to you now, right? Okay, history was, and it will always be, one of my true loves. And when I graduated, everyone told me I should be a teacher, and I always said, not a chance. Kind of funny, huh? So fast forward a few years, and I'm getting kind of bored in the museum that I worked in, so I went back to college for a complete career change and landed in the lab. Now, I love history, but I love lab even more. So I spent several years in the lab, and after a while, I had the urge to go back to school. So I did, and I came out with a master's in public health. And somehow, some way, here I am teaching because life is funny like that. I really love teaching. I have really enjoyed what I do, and I love teaching science and lab sciences. So I hope that you guys will enjoy my classes. Anyway, long story short, I believe that knowing the history of things is very, very important. So knowing where we came from can help us get where we are going. And that is why we're going to talk about the history of molecular. And also, because Opal and her tongue shouldn't get all the attention, here's my rescue kitties, Cricket and Milligram. They're just too much, aren't they? Okay, first up we have Gregor Mendel. So Gregor Mendel was an Austrian monk who studied heredity in pea plants. He discovered the basic laws of inheritance and noted that inheritance of each trait was determined by what he called units or factors. Now these are known as genes. So he also determined that individuals inherit one such factor from each parent. And Gregor Mendel is today considered the father of modern genetics. Frederick Meister. So he is probably one of my favorite scientists in this whole lecture. So this guy legit took dirty bandages out of the trash and used the pus from those dirty bandages for his studies. Now, one has to wonder if this required informed consent, although probably not. Anyways, Meister was actually smart to use the pus because pus is made up of what? That's right, white blood cells. And what do white blood cells have that red blood cells don't have? A nucleus. Good job. And from this nucleus, Fred was able to isolate a compound that he called nuclein. So at this time, scientists thought nuclein functioned as a storehouse for the phosphorus atom. But today, knowing what we know, he was likely working with nucleic acids. So he was really the first one to isolate what they called nuclein, but we know now as nucleic acids. So next up we have Frederick Griffith. In 1928, he began experimenting with pneumococci strains in mice. So he used one pathogenic strain and one non-pathogenic strain of pneumococci, injected them into mice, and from the results determined that transformation was actually possible. So here we have a picture of strep pneumo gram stain. You can see the gram-positive cocci. They're in pairs and they're kind of lancet-shaped. Hint, hint for your micro course. And then here we have an image of Frederick Griffith. And I mean, honestly, who doesn't love a scientist that poses with their super cute doggo? So a little more detail on Griffith's transformation experiments. So the two strains of pneumococci he used were the rough strain, or R strain, which was unencapsulated and non-pathogenic.
This is something that we know.
And the smooth strain, or S strain, which was encapsulated. So in the 1940s, a trio of scientists picked up where Griffith left off. So we have Oswald Avery, Colin MacLeod, and Macklin McCarty. And they worked together to determine that DNA was required for transformation. So Griffith figured out that transformation was possible, and these three were the ones that determined that DNA was required for that transformation. So at the time, what was well known about DNA was that it was much simpler than proteins, and many scientists still thought that proteins coded for genetic material. So when Avery, McLeod, and McCarty released their work, many of these scientists were still skeptical. They thought that DNA was simply too simple to code for the genetic material. So what exactly did they do to determine DNA was a transforming factor? Well, they essentially took Griffith's heat-killed smooth strain of pneumococcus and removed lipids and sugars, leaving only proteins, RNA, and DNA. So then they treated this solution with different enzymes. So the enzymes would destroy either a protein, RNA, or DNA. Next, they added the rough strain to the heat-killed smooth strain to see if transformation occurred. The smooth strain appeared in the solution that contained RNA and DNA, and the solution that contained protein and DNA, but it did not appear in the solution that contained no DNA. So this result led to the conclusion that transformation requires DNA. Therefore, DNA is the genetic material of the cell. So some time passes after Avery, McLeod, and McCarty, and many scientists work on many different experiments and end up with the basic composition.
This is something that we have also done in our basic course GCMB 203, right? So this is the base, this is the pentose sugar, this is the phosphate. It's going to link into two other phosphates that will form the this thing, triphosphates, right? So this is the phosphodiester bond. This is the hydroxyl group that is found in DNA, and because this has no oxygen, that is why this is called as a deoxy. In our case of RNA, even 2' position will have a hydroxyl group. Right? This nitrogenous base can come in the form of A, T, G, or C, and in the case of RNA, T is replaced by uracil.
Composition of DNA. So we know that DNA is made up of a sugar, which is deoxyribose. Erwin Chargoff was a chemist, and he only got interested in DNA after reading about Avery, McLeod, and McCarty's report identifying DNA as a hereditary material. Remember, many scientists were skeptical of their report, but he actually believed them from the very beginning. And in 1949, he determined the base composition of DNA varied between species. And then he discovered that there was a fixed ratio of bases in DNA. So the amount of thymine was equal to the amount of adenine, and the amount of cytosine was equal to the amount of guanine. Now, this led to what is known as Chargoff's rules. And the rules are, in any species, the ratio of A to T is 1:1, and G to C is also 1:1. And also, other ratios such as A to G will vary from species to species. So he made several very important discoveries. And it's important to note that while he did determine that the amount of A equals T, the amount of C equals G, he didn't actually suggest that those were always paired together, though it was kind of hinted at. So Rosalind Franklin, we've all heard of her. She was a Cambridge-educated chemist, and she eventually ended up in Paris, where she learned X-ray diffraction techniques, which she then took with her to King's College in London. Franklin was able to set up and improve the X-ray crystallography unit at King's College, and Maurice Wilkins was already working there. So Franklin was actually hired while Wilkins was on leave, and when he returned, he assumed that Franklin was his assistant. He treated her as such and was not really told otherwise, and this really set the stage for what was a very tumultuous relationship. So Rosalind was the one who was able to get high-resolution photos of crystallized DNA fibers. So from her images, she was able to determine the basic dimensions of DNA strands and that the phosphates were on the outside of what she thought to be a helical structure. Now Wilkins, who was working with Franklin, he was a physicist educated at St. John's College in Cambridge. Now, his early career, he actually worked on improving radar and then was part of the Manhattan Project, which resulted in the atomic bomb. Now, after his work on those war projects, he began studying biological molecules like DNA and viruses. Eventually, he was also able to produce X-ray diffraction images, and along with Franklin's images, they were able to help determine the shape of DNA. Ultimately, it was James Watson and Francis Crick who proposed a 3D model of DNA in 1953. And their original model was inside out, but eventually they proposed the model that we know today. Rosalind Franklin's X-ray diffraction images were key to getting the model right. So Maurice Wilkins also contributed some images. In 1962, the Nobel Prize for Physiology or Medicine was awarded to Francis Crick, James Watson, and Maurice Wilkins. And the Nobel Prize was not awarded posthumously, so Rosalind Franklin, who had died in 1958, was not listed among the winners. However, her contribution is more widely recognized today. And it's important to note that in 1953, Rosalind Franklin and Maurice Wilkins actually published papers in the same issue of Nature where Watson and Crick first published their findings. So they actually all had really groundbreaking work published in the same journal in 1953. So this is a quick history of...
So even today, Nature continues to be one of the topmost journals, right, for scientific publications. Okay, and it's a tough journal to publish your scientific findings in. Okay, there are a few other journals that are like that, but it also depends on the quality of
Grammarly.
Okay, so many factors are there, but Nature continues to be one of the topmost journals. We'll quickly have a look at another short.
Does more than catch errors. With Grammarly, you can find really good.
You're watching a genomics education module produced by the Division of Genomic Medicine at UC Davis. This module will focus on different tests used in molecular genetics, how they're different, and when to use each one. Let's start by talking about the broad categories of molecular diagnostic test types. If you suspect that an individual might be affected by a chromosomal abnormality, you'll need to do a cytogenetic analysis. Chromosomal abnormalities include issues like an extra or missing chromosome, as in trisomy 21, or extra or missing parts of chromosomes. There are a few different options for cytogenetic analysis, which will be discussed in a different module called cytogenetics. If you want to zoom in on a chromosome and take a closer look at the sequence of DNA bases, you will need to do a molecular genetic analysis. However, in the quickly changing field of genetics and genomics, cytogenetics and molecular genetics continue to overlap more and more. Let's say that you've decided that you want to run a molecular genetics analysis. Now you'll need to consider which test to use. There are a variety of options. To determine which is the best test for your particular case, you'll need to think about which part or parts of the genome and how much of the genome you need to assess. For example, if you want to look at all nucleotides in the entire genome, you can order whole genome sequencing. For a human, this means sequencing all 3 billion base pairs in the human genome. The whole genome includes coding regions like exons, as well as non-coding regions such as introns and regulatory elements. Here are a few common clinical indications for whole exome sequencing: diagnostic odyssey, many differential diagnoses, atypical disease presentation, or disorder with a high degree of genetic heterogeneity. Another option is whole exome sequencing, or sequencing of just the exome. Whole exome sequencing captures just the parts of DNA that code for the mature RNA strand. Common clinical indications for whole exome sequencing are similar to those for whole genome sequencing. It is important to keep in mind, however, that whole exome sequencing...
So in the case of whole genome sequencing, you will be sequencing the entire 3 billion nucleotides, right? So that is called as WGS, or whole genome sequencing. In the case of whole exome sequencing, all you need to do is to take the messenger RNA, right, significantly less, right, convert them into cDNA and then clone them and start sequencing, right? So it is probably going to be, if you remember your genomics class, it's literally 98% of your genomic sequence has, all these introns, regulatory sequence, promoters, upstream regions, downstream regions, and all that. Your coding region is only about 2%. So it is, significantly, much more, comparatively much more cheaper to do whole exome sequencing. The only issue is if there is a promoter mutation, right, or if there is an intron-exon boundary mutation in that, then we are going to miss that. Otherwise, by and large, if it's a variation in the protein, you will be able to catch it in whole exome sequencing. Okay.
Does not provide any detailed information beyond just the protein coding regions of each gene. To get that information, whole genome sequencing is required. Another option is targeted mutation analysis. This is useful when you already have a suspected condition or group of conditions in mind and know where the associated gene is located. For example, if you are testing a patient for sickle cell anemia, there is no need to sequence more than one specific locus. Targeted mutation analysis is usually used for individuals who have an affected family member with disorder who has already had genetic testing and a specific mutation in a gene was identified. These individuals would then be tested for the same mutation in the same targeted gene. In some cases, you may have a hypothesis for a condition or group of conditions you'd like to test for. If that condition has locus heterogeneity, meaning that it can be caused by multiple genes, you may want to consider ordering a panel test. For example, many genes can contribute to breast cancer, so a breast cancer panel may sequence a dozen or more specific genes that are known to be associated with breast cancer. Panel testing is usually done when a patient has a specific condition that can be caused by a number of different genes, such as breast cancer or hypertrophic cardiomyopathy. So to recap, there are many different options for molecular genetic testing. The most important question to answer before deciding on which of these options to go with is how much of the genome you need to analyze. The sequencing method of choice today is next-generation sequencing, or NGS. NGS utilizes massively parallel, high-throughput technology to produce large-scale automated genome analyses. This is much faster and cheaper than the original method, Sanger sequencing. So to recap, there are 4 main test types available for DNA analysis at the level of the nucleotide: whole genome sequencing, whole exome sequencing, targeted mutation analysis, and panel testing. If you would ever like help deciding which is the most appropriate genetic test to order for your patient, the Division of Genomic Medicine is here to help. Our clinic is...
In the current COVID times, how molecular diagnostics have helped, we know that RT-PCR has now become the gold standard. So let's quickly watch this short video on COVID diagnostics.
Here at the UCSC Molecular Diagnostic Lab, we conduct PCR tests on swab samples to determine whether the sample contains SARS-CoV-2 coronavirus, which causes COVID-19.
The MDL is a nonprofit testing lab dedicated to assisting in areas of need, including the uninsured or essential workers, and serving the UC Santa Cruz population.
Our goal is to facilitate a return to education, learning, and research, and to support economic and community recovery. The idea for this lab was built in March 2020. By May 1st, we received approval from the state to start testing. We test samples that we receive from our partner medical providers. Our partnerships are aligned with our mission and commitment. We have partnered with federally qualified centers such as Santa Cruz Community Health and Salud para la Gente, which serve all populations, including agriculture workers, homeless and low-income families, and with the county to serve emergency testing needs.
Through Santa Cruz County Public Health, we are piloting testing with several hospitals and organizations essential for Santa Cruz's safety and recovery.
Our expectation is that we will continue to provide such emergency testing through the pandemic.
Let's start with a few terms. A Qpcr stands for Quantitative Polymerase Chain Reaction and is a technology used for making copies of DNA and RNA. By doing this, the quantity of DNA, or in this case RNA, can be measured. Testing on a Qpcr device is consistent with the Centers for Disease Control, or CDC, approach. In fact, our test is based on the same principle as a CDC test, but uses different probes to detect the virus.
This assay builds on the Qpcr assay developed by Columbia University and modifies it to use reagents that are not used widely, therefore avoiding reagent supply problems. The assay also uses automation at every key step, from RNA extraction, reagent dispensing, and Qpcr. An assay is a test of a substance to determine its components, and it's frequently used to test for the presence or concentration of infectious agents. Reagents are substances added to a system to cause a chemical reaction or to test if a reaction occurred. Through rapid innovations, the MDL has been able to move from the manual process shown to scale testing and return results within 24 to 48 hours without sacrificing sample integrity and quality.
Before entering the lab, all personnel must wear personal protective equipment, otherwise known as PPE, and must be trained on biosafety and protected health information. The PPE required includes N95 mask, lab coat, goggles, and gloves. This is to protect the lab staff and to ensure that there is no contamination in the lab. Any small amount of DNA from personnel can compromise results and change effectiveness of the testing. The protected health information training is directed to ensuring patient privacy and is part of regulatory rules.
We use the robot to dispense RNA Shield, which is a reagent that inactivates the virus so it is no longer a threat to the lab, staff, or anyone touching the sample. RNA Shield is able to provide this protection while maintaining the virus's genome. That way, we can safely test for the presence of the virus. RNA Shield is dispensed into 1.4 milliliter 2D pre-barcoded tubes. Etched barcodes allow us to process samples in a high throughput manner to instantly know which samples are coming back to us. The test kits are built in-house in our lab. The kits can be assembled in minutes using our Agilent Bravo, which is a 96-well liquid handling robot which can very accurately dispense liquid. The kits include tubes filled with RNA Shield, racks, and swabs. Once kits are assembled, they're sent to providers to collect patient samples. The providers take one of these tubes and scan the tube's barcode to register the patient. A swab will then be conducted. They return the kit to us by courier.
Once kits arrive back at our campus, they are brought to the lab and sterilized in an oven at 70 degrees Celsius. This is a safety measure designed to ensure that the virus is deactivated and the genomic material is maintained. Once the kits are sterilized, they are inserted into a barcoded rack, which is placed on a scanner. Then up to 96 tubes can be scanned all at once. The scanner automatically registers and uploads their data into our Laboratory Information Management System, also known as the LIMS. The LIMS tracks all samples scanned all at once, checking for personal health information to be sure it matches. Because of this automation, we have been able to scale up in order to process many patient samples all at once.
Once samples are registered, we can begin our automated workflow to extract the sample's RNA material. This is when we begin the process of the Qpcr assay. PCR is able to exponentially amplify the virus so that it can be detected even if present in low quantities. Our assay can detect less than a single viral genome per microliter of swab material. To build this assay, we took primer and probe sequences that recognize SARS-CoV-2 developed by Columbia University. We then modified the RNA extraction method and reagents to avoid reagents that are in limited supply. Finally, we implemented the assay in a 96-well plate format so that 93 samples and 3 controls can be processed simultaneously using our robots. Once those samples are run on a thermocycler, the data is outputted. We load a positive control to ensure that the reagents are working as planned and we can detect the virus. Whether a sample reaches that threshold or is below that threshold determines whether the patient is positive. In this image showing data output, there are 3 wells highlighted, depicting one particular sample. In this case, RP is the internal control that tells us that we've collected enough material. N1 and N2, which are the purple and fuchsia colors, measure the presence of the virus. N1 and N2 are amplified in the sample, indicating that the patient has COVID.
A clinical laboratory scientist will review the data output and assign a result to every sample. In addition to detected and not detected results, the sample can be of poor quality and the result will be reported as quality unsatisfactory. Another type of result is inconclusive, when we cannot tell whether the virus is present or not. This could happen if the virus is present at a minuscule level or the patient has another virus that is very similar to SARS-CoV-2.
Usually, if samples come back positive, the patient is notified within 24 hours. All results are returned to the provider within a 24 to 48-hour time frame.
Our expanded testing capacity has been made possible in part through funding from the Coronavirus Aid, Relief, and Economic Security Act, support from Santa Cruz County.
Okay, so you got a flavor for what molecular diagnostics is all about, right? So we'll continue our journey on molecular diagnostics in our class tomorrow. Okay, so this is just an overview. This is just an overview. Okay, and as we saw, molecular diagnostics has now become the buzzword, right? People and governments and pharma companies are encouraging people to come up with innovations so that we can catch these diagnostic kits. We can make these diagnostic kits affordable and cheap. Okay. Any questions that you may have? Okay. If there are no questions, then we will meet in tomorrow's class. All right? Okay. Thank you. Bye.